Finally, the third point to understand involves a more macro-level explanation of the thermal field in tumors, which relates to Gibbs free energy. The theory of Gibbs free energy was initially proposed to describe whether a chemical reaction can spontaneously proceed. Those familiar with natural sciences know that the universe continues to develop either through an increase in entropy, where order transitions to disorder, ultimately leading to a state of thermodynamic equilibrium, or through a decrease in the system's energy, releasing heat in processes like aerobic respiration, which converts glucose into energy stored as ATP. Gibbs free energy, delta G, is central to understanding these processes. For a reaction or metabolic process to proceed spontaneously, it must either increase entropy, delta S, or decrease the system's total energy, delta H. In aerobic respiration, a significant amount of heat is produced, resulting in a considerable reduction in the system's internal energy, delta H, with only a slight increase in entropy, delta S. Conversely, anaerobic respiration, fermentation, releases much less energy, implying a smaller decrease in internal energy. Thus, it compensates by generating a greater increase in entropy. In tumor tissues, this increased entropy manifests as a more disordered state compared to normal tissues. For the organism, this rise in entropy leads to various pathological conditions. The heat generated by tumors is also a direct consequence of this entropy increase. As entropy rises, the surrounding heat also increases. The fourth point to consider is the central control system inherent in normal biological cells. Evolutionarily, this system has adapted to align with circadian rhythms, regulated by a set of clock genes within our DNA. These genes function like an internal clock, controlling various biological rhythms such as sleep-wake cycles, sexual desire, and energy metabolism. In tumor cells, these clock genes undergo primary or secondary mutations, disrupting their normal heat production rhythms. While normal cells might produce more heat during the day and less at night, tumor cells might exhibit the opposite pattern, resulting in altered heat production rhythms. By using sensors to monitor these changes in heat production rhythms, we can identify potential tumorigenic changes in tissues. For example, consider a graph generated by a biorhythm monitor system showing temperature variations over 1,200 minutes, 20 hours, for both breasts. The blue line represents the temperature of the breast with breast cancer, while the orange line represents the temperature of the normal breast. The blue line consistently shows higher temperatures and less smooth temperature fluctuations, indicating altered heat production rhythms. Additionally, the sensor includes a humidity monitor to compensate for temperature sensitivity. Higher skin temperatures induce sweating, leading to higher overall humidity levels and more significant humidity fluctuations on the side with breast cancer. The fifth point addresses how tumors stimulate lymph nodes, crucial immune organs involved in tumor metastasis. Clinicians know that lymph nodes play a vital role in the body's immune response. When a tumor begins to metastasize, it first encounters natural killer (NK) cells. If the tumor evades these cells, it may enter the lymph nodes, proliferate, and generate heat. This proliferation also activates a series of immune and inflammatory responses within the lymph nodes, further increasing their temperature. If the lymph nodes fail to contain the tumor, it may spread to distant body parts, marking advanced cancer stages. For breast cancer, the axillary lymph node group is particularly significant due to its anatomical proximity to the breast. The sentinel lymph node, located near the upper outer quadrant of the breast, serves as the first line of defense against breast cancer metastasis. Detecting heat in this sentinel lymph node can indicate early-stage breast cancer, stage 2, providing a critical monitoring point for breast cancer diagnosis. Breast cancer metastasis is closely related to a specific anatomical knowledge that many of us can learn about. The tumor cells inside our breasts typically migrate towards the group of lymph nodes under our armpits. These armpit lymph nodes are mainly located at the outer upper quadrant of our breast. Within this armpit lymph node group, there is a particular node called the sentinel lymph node. Sentinel means guard or watchman in English. Therefore, this lymph node serves as the first line of defense against breast cancer metastasis. If the tumor reaches our sentinel lymph node, it usually indicates that it has progressed to stage 2, which is considered an early stage of breast cancer. If, at this stage, we detect that the sentinel lymph node is inflamed, there is a high probability that it is a stage 2 breast cancer. The sentinel lymph node is located in a relatively fixed position, near the nipple at the outer upper quadrant area.